Hey folks, my name is Sean Thomas, and I'm a PhD student at the University of Missouri. Today I'm going to talk to you about the Mustard Mayhem, where I'll go over my work to place and date the whole genome triplication in the tribe Brassiceae. For those not familiar with this group, tribe Brassiceae is a monophyletic group within the mustard family that contains everyone's favorite vegetables like broccoli, cabbage, kale, and others. However, these mustards are quite messy. They have many nested polyploidy events, including AT beta, which is this Brassicaceae whole genome duplication, AT alpha, which occurred at the base of the family Brassicaceae, and finally, this Brassiceae whole genome triplication, which is going to be the focus of this presentation. So we have this ancient hexaploidy, or whole genome triplication in the Brassiceae, but we're unsure of where and when it occurred. Previous data shows that it shared amongst the core Brassiceae, shown here. However, we are unsure if earlier diverging lineages share this whole genome duplication. And we're interested in understanding the timing and placement of this polyploid event, especially because polyploid-derived genes have been associated with interesting traits such as domestication. Since polyploidy plays a role in domestication, another question I'm interested in is, does it influence other traits? Catherine and Tasha are awesome undergrads working with me to explore how polyploidy may play a role in abiotic stress tolerance. So feel free to scan the QR code on the page or take a picture of this to find out more. So first, I'll go over my work to place the Brassiceae whole genome triplication. So looking at some previous evidence from comparative chromosome painting, this revealed triplicated Arabidopsis segments in Brassica and Cacaili. As you can see here, Brassica nigra, Brassica rapa, and Cacaili have triplicated segments that were derived from Arabidopsis. So this gives us some evidence that the tribe Brassiceae, or at least these three species, have an additional polyploid event from the AT alpha event, or the Brassicaceae whole genome duplication. So looking at the phylogeny, we have evidence for the whole genome triplication at these 3 points. So based on this, we can assume that the whole genome triplication is conserved in this clade from Cacaili up, or within this core Brassiceae group. A more recent study assembled a chromosome scale genome for Brassica rapa and found more evidence for the whole genome triplication. Based on the data in the paper, the authors hypothesized that this whole genome triplication would have occurred in 2 steps. The first step, or this initial whole genome duplication, would be this initial tetraploidization, followed by a subsequent diploidization, which would yield segments from 2 genomes in its descendants. The second step would then be another whole genome duplication, following that first diploidization event. So the second step is hypothesized to be an allopolyploid event. And then after this event, we would see another diploidization, which would yield 3 genomes or segments of 3 genomes, similar to what we see in the chromosome paintings from the previous study. And so looking at this whole process over time, we have this 2-step process, which first produces the first set of 2 genomes, and then the additional polyploid event, which results in our triplicated genome or hexaploid genome. So in order to place the whole genome duplication, I'll use 2 lines of evidence. The first line of evidence would be to compare KS plots across the phylogeny. And then the second would be a phylogenomic approach to query duplications in gene trees and map those back to the species tree. So looking at my workflow here, I started with 53 RNA-seq samples, assembled those as transcriptomes, and then from those transcriptomes, I built gene trees using IQ-tree, and also built a species tree using AstralPro. And so far, what I've been able to do is conduct this fast KS analysis, which will allow me to produce KS plots. What's still in the works is running this PUG analysis, which is the phylogenomic approach to query those duplications in gene trees and map them to the species tree. So for those unfamiliar with KS plots, they are distributions of the frequency of synonymous substitutions, or KS, in putative paralogs. So looking at this figure here, across the x-axis, we have KS. As paralogs diverge over time, we expect that they will have a higher rate of synonymous substitutions. When a polyploid event occurs, we expect to see a peak in KS indicative of that event. So in this figure here, this shows the KS plot of Brassica rapa. And there's a peak for the family Brassicaceae, or this AT alpha event here. And then this peak here is indicative of the Brassiceae mesohexaploidy. The authors of this paper set these boundaries for the Brassiceae mesohexaploidy as these dotted red lines, And so we'll use those in our data to see if we see consistent results. So on the right here, we have a sample from my data set, and this peak here is consistent with the Brassiceae mesohexaploidy. So here on this figure, I have the KS plots for my samples shown as these ridgelines across the phylogeny. Taxa from Vela upwards that are colored are part of tribe Brassiceae. So if you squint real hard and look at these ridgelines, you can see that all of the taxa from Vela upwards share this peak at the hexaploidy and are within the dotted red lines or the bounds that were set from the Brassica rapa paper. And starting here at Auricophragmus, that's this ridgeline here, we see that it doesn't share that Brassiceae peak, and the KS plots become more variable in their topology. If you weren't convinced from the ridgeline plots, I've also overlaid the KS plots onto these figures here. The KS plots in purple are in-group taxa, the Brassiceae, and all the KS plots are overlaid on top of each other. The same is happening here in this panel. The green KS plots, these are our outgroups. So this shows us that the Brassiceae taxa have this peak that's consistent with the Brassiceae hexaploidy event, whereas the outgroups are much more variable in where we see KS peaks. Some seem to have peaks that are consistent with the Brassiceae hexaploidy, but it's likely that these lineages have their own polyploid event that may have resulted in this pattern. And finally, I'd like to point out this panel here. And so we have our in-group taxa here, our out-group taxa here, and what we're looking at here is the median KS values. And our in-group taxa fall nicely within that middle bound, whereas the out-group taxa are spread out much more variably. And so these KS plots suggest that the Brassiceae hexaploidy must have occurred somewhere along this branch leading to the tribe Brassiceae. So previously I showed you chromosome paintings just within the core Brassiceae, but here's what we see in the sister group to the Brassiceae in this Auricophragmus lineage. And so we see Auricophragmus is sister to the tribe Brassiceae, and we see that it contains duplicated segments of Arabidopsis. So this leads me to wonder, did the initial whole genome duplication, or step one of the hexaploidy, occur on this branch leading to Auricophragmus? And so if this were the case, what we would see is that step one would occur on this branch leading to Auricophragmus, and step two, the secondary whole genome duplication, would be occurring on this branch leading to the Brassiceae. Another possibility is that both of the steps occurred on the branch leading to the Brassiceae. And based on our FastKS data, that's all we can conclude, is that the Brassiceae hexaploidy occurs along this branch here. However, the KS plot data alone, I don't think provides enough evidence to only place the hexaploidy along the branch leading to the Brassiceae. I think we would need to have chromosome scale genomes in order to confirm this. So now switching gears, I'd like to go to the second part, which is dating the Brassiceae whole genome triplication. And so when we're thinking about dating the Brassiceae, we have uncertainty in previous Brassiceae divergence time estimates. Previous studies have shown that the tribe diverged from the rest of the family Brassicaceae from 12 to 28 million years ago. And we have some uncertainty due to questionable fossils in the Brassicales. So looking at previous divergence time estimates, we see, right, they range from 24 to 12 million years, and right, they're spread out all over. And so the approach that we want to do to avoid this problem is using this approach from Walden et al. 2020, this paper here. So they did a divergence time analysis of the Brassicaceae, and they were able to avoid contentious Brassicales fossils by using more robust fossil calibrations nested within the rosids. So in order to do this, first we need to infer a phylogeny with the rosids as an outgroup, or with some rosid taxa as an outgroup. And so because this will be a lot of taxa, we're using chloroplast data in order to do this. And then secondly, the second step would be to then conduct our divergence time analysis using the program BEAST, which implements a family of MCMC algorithms for various evolutionary analyses. And so looking at our workflow here, we have 216 samples that come from genome skimming data or existing chloroplast data across the rosids, but with a focus on the Brassicaceae taxa. After that, we assemble and align our sequences. So what we did was we used the Brassica rapa chloroplast coding sequences as a reference, and then used spades to assemble, and then math to run the alignments. And then finally, what we're doing is a divergence time analysis. So we're using IQ-tree to get a maximum likelihood phylogeny, and then beast to run the actual divergence time analysis using the fossil calibrations and the rosids. And so we haven't quite finished the divergence time analysis, but we do have some preliminary results. And so looking at the Brassiceae node, so this node here, we see a mean age of 10.6 million years, and this 95% HPD interval of 8.6 to 12.7 million years. And then if we look at our Auricrophragmus node, we have a mean age of 12.1 million years, and the 95% interval at 9.9 to 14.5 million years. And in this node, we have a slightly lower posterior probability. And so placing, so just looking at the Brassiceae again, looking at the divergence time estimates, we see that our Brassiceae divergence time estimate is much younger than any of the previous estimates. And given that these are preliminary results, they may change as the program continues to run, but we'll see when the results actually come back. And so some conclusions from my talk, the FastKS data, or the KS plot data, suggests that the Brassiceae whole genome triplication occurs on the branch leading to the Brassiceae as currently circumscribed. And from our divergence time, our preliminary results from our divergence time analysis suggest a much younger age for the Brassiceae than previously shown. So future direction. So well, next steps would be for me to finish the PUG analysis in order to place duplications from gene trees onto the phylogeny, and then also for that divergence time analysis to finish up. Some future directions would be to have additional genomes or chromosome scale genomes, and then using synthetic analysis to help resolve this two-step Brassiceae whole genome triplication. And so finally, I'd like to just give some acknowledgements to my collaborators shown here, Dr. Sean Abrams, Dr. Tatiana Arias, Dr. Nora Walden, Dr. Alex Harkis, Dr. Jacob Washburn, and Dr. Chris Pierce. And I'd like to thank the Washburn Lab for review and feedback for this talk. And then here are my funding sources. And so with that, I'll take any questions.